So during our last lecture, we examined the eukaryotic cell parts known as the, the eukaryotic cell parts called microbodies. And basically that we said microbodies are structures that have oxidative components to them. These would be things that would have enzymes that are involved with the oxidation of fatty acids. So a variety of enzyme-bearing membrane-bound vesicles. They contain enzymes involved in the oxidation of fats. They also have catalase, which is an enzyme that will break down a poison to our cells called hydrogen peroxide, H2O2, into water and oxygen gas. Vacuoles themselves are membrane-bound structures in plants. Various functions of vacuoles depending on the cell type. Overall, there are different types of vacuoles. Of course, there are the central vacuole in plants. We'll look at that a little bit. And then in some fungi and protists, you have things called contractile vacuoles. Some of those will play a role in osmoregulation, regulating water. And then there are also storage vacuoles for storage of substances within inside the cell. Of course, there you see the central vacuole. The central vacuole makes up the bulk volume of a plant cell. So here you see a typical plant cell. And you see that central vacuole right here. The membrane to the central vacuole is called the tonoplast. And what happens, the membrane surrounding the vacuole, the tonoplast, has channels that allow water in and out to maintain plant cell tonicity or osmotic balance. So when the plant cell is filled with water, that water being of course stored in the central vacuole, that's where we are going to say that the plant is exerting turgor pressure onto the rest of the plant cell. The vacuole is also thought to store enzymes that are involved in the breakdown of macromolecules and those used in detoxifying foreign substances. The other thing that the vacuole does, in addition to water balance and breaking things down, we do know that it is used for storage of useful molecules like sugars, ions, and even some pigments can be stored in the vacuoles. Then there is the mitochondria. Mitochondria and chloroplasts have many common structural and functional similarities. So the mitochondria itself, it's found in all types of eukaryotic cells. It is bound by membranes. There is an outer membrane, and then you have an inner membrane, and if you recall, that inner membrane is folded to form structures called cristae. So they would be the cristae of the mitochondria, or if you call it singular, it would be crista. A mitochondrion, then, if you look between the outer and inner membrane, you have that intermembrane space. And then, in addition to that, inside the inner membrane, the innermost compartment of a mitochondrion is the matrix. Mitochondria are really cool because they do have their own DNA. Every time that the cell does divide, it does not replicate mitochondria. The mitochondria will divide themselves, but they do need the help of a nucleus of a cell in order to do that, because the nucleus will send out DNA, which will be translated and transcribed into proteins, which will then kick into play for mitochondrial replication. So mitochondria do have their own DNA, and the DNA itself does contain several genes that are going to produce proteins essential to the mitochondrion's role in oxidative metabolism. Mitochondria, in many aspects, act as a cell within a cell because it does contain its own genetic information and specifying proteins for unique functions. Also, a eukaryotic cell, as I said, does not produce its own mitochondria. The mitochondria will reproduce themselves, but you do have to have the help of the nucleus in there. If you look at the structure itself, the mitochondria, you have the outer membrane. Here's the inner membrane, and it's folded to form those crista. And then you have the intermembrane space. The intermembrane space is going to contain enzymes. And there you could see the mitochondrial circular DNA in there as well. Also in there you have ribosomes, because ribosomes are going to be used to make those proteins. So here you see a picture of what one of those mitochondria will look like. Over here you see an electron micrograph. So essentially what the mitochondria does, it is going to be that organelle where cellular respiration is going to take place. So it's going to produce energy from the cell. On the other hand, we have the chloroplast, and chloroplast also very similar to mitochondria. These are organelles present in the cells of plants and some other eukaryotes. They contain chlorophyll, which is used for photosynthesis. Again, they are surrounded by two membranes. You have an outer membrane and an inner membrane. And you also have inside, you have thylakoids, and the thylakoids are membranous sacs within the inner membrane, and a stack of thylakoids is called grana. And then, of course, the area in which you find those thylakoids is in a space called the stroma. Again, chloroplasts do have their own DNA, but many of the genes that specify chloroplast components are also located in the nucleus. Some of the elements used in photosynthesis, including those proteins that are specific protein components necessary to carry out the reactions, are synthesized entirely within the chloroplast itself. So here we do see a structure, and of course this would be the site of photosynthesis. So photosynthetic reactions take place. That'd be the light-dependent reaction occurring in the thylakoids, and then the light-independent reaction would occur in the stroma. If we look, basically looking at the free-living cells at one point, like the mitochondria and chloroplast, there are some other organelles that also have DNA in them. So these other DNA-containing organelles in plants are called leucoplasts. Leucoplasts lack pigments and are complex internal structure. In root cells and some other plants, the leucoplasts serve as starch storage sites. Leucoplasts will store starch, amylose, and sometimes termed amyloplast. These organelles, like chloroplast and leucoplast and amyloplast, are collectively what we would call plastids. But overall, we do come to mitochondria and chloroplasts have arose by this thing called endosymbiosis. And endosymbiosis itself, if you look, we would say that the theory of endosymbiosis proposes that some of today's eukaryotic organelles evolved by a symbiosis arising between two cells that were each free-living. One cell will be a prokaryote, was engulfed and became part of another cell, which was the precursor of what we would today consider the modern eukaryotic cell. According to this theory, the engulfed prokaryotes provided their host with certain advantages associated with their special metabolic abilities. The two key organelles, the two key eukaryotic organelles that are believed to be descendants of the endosymbiotic theory of endosymbiotic prokaryotes are the mitochondria, which are thought to have originated as bacteria capable of carrying out oxidative metabolism, and the other one being the chloroplast, which arose from photosynthetic bacteria, and we will learn about that a little bit later. So if we look here you can see here's an unknown archaeon that would come in to become the nucleus there. The protobacterium would be the oxidative bacteria, which became the mitochondrial organelle, and the cyanobacteria, which would be able to carry out photosynthesis, has been engulfed in and eventually became the chloroplast. So now we have our modern eukaryotic cell there. The cytoskeleton, basically it's a network of fibers, protein fibers, found in all eukaryotic cells. It's a dynamic system that is constantly forming and disassembling. Basically, when you look at the network of protein fibers, it supports the shape of the cell and it keeps organelles in fixed locations. The three types of fibers that we do find in the cytoskeleton itself are the microfilaments or actin filaments. These are going to be two protein chains that are loosely twined together. The actin filaments are made of two strands of globular protein actin that are twisted together. They are often found in bundles or in branching networks. The actin filaments in many cells are concentrated below the plasma membrane in bundles known as stress fibers, which may have a contractile type function. So there we see movements like contraction, crawling, and pinching. Then we have microtubules. Microtubules are composed of alpha and beta tubulin protein subunits. And they're going to be arranged in a side-by-side to form a tube. Microtubules are comparatively stiff cytoskeletal elements, and they have many functions in the cell, including intracellular transport and the separation of chromosomes that will happen during mitosis. And then lastly, you have those called intermediate filaments. And intermediate filaments are composed of overlapping, staggered tetramers of proteins, and these tetramers are then bundled into cables. And this molecular arrangement allows for a rope-like structure that is going to impart tremendous mechanical strength to the cell. So it's very stable and therefore not able to be broken down. Intermediate filaments are typically 8 to 10 nanometers. When you look at microtubules, they're about 25 nanometers. And actin filaments, they're about 7 nanometers. So here you can see a picture of what the actin filaments look like. I just discussed their structures. Yeah, the microtubules right here, and then the here you can see how those cables are formed in the intermediate filaments. Centrosomes. Centrosomes are regions surrounding centrioles in almost all animal cells. Basically what's going to happen is microtubule organizing center. This is where microtubules are going to come together. They can nucleate, and the animal cells and most protists have centrioles or these pairs of organelles. Plant cells and fungi usually lack centrioles. But if we look at the centriole itself, here it is. Centrioles are barrel-shaped organelles found in the cells of animals and most protists. They occur usually in pairs and usually located at right angles to each other near nuclear membranes. The region surrounding in the pair is almost all animal cells is referred to that centrosome. So surrounding the centrioles in the centrome is the pericentriolar material, which contains ring-shaped structures composed of tubulin And basically what's going to happen is the pericentriolar material can nucleate the assembly of microtubules in the animal cell. And basically what will this do is this will help play a role in cell division. So centrioles will play a role in cell division by shooting out that spindle fiber. And you know that the spindle fibers will then go attached to the chromosomes at the place called the kinetochore. The cytoskeleton also plays a role in cell movement. Essentially, all cell motion is tied to the movement of actin filaments, microtubules, or both. Some cells crawl using actin microfilaments, and then you have the case of the flagella. The flagella and cilia have a 9+2 arrangement of microtubules. Not like prokaryotic flagella. In the eukaryotic cell, that 9+2 arrangement is going to create the flagellum, and the cilia are shorter and more numerous than one or two long flagella. If you look here, here is that 9+2 arrangement. So you have 1, 2, 3, 4, 5, 6, 7, 8, 9, and 2. So you got that 9+2 arrangement, microtubule pairs, and then central microtubule pair right there. So here you have that radial spoke, and the dynin arm is right out in here. So if you look at the part of the flagellum itself, here is that structure that you're looking at here. And then you have the plasma membrane that would surround it. And there would be your basal body. So there's the microtubule triplet there. I want to quick go back to the, I'll come back to that. All right, last thing for today, if we look at the eukaryotic cell wall. Eukaryotic cell wall, we have the plant cell wall. It's going to provide protection and support. Eukaryotic cell walls are in plants, fungi, and many protists. It is different from the cell wall that we see in prokaryotes. Plants and protists, the major component of the cell wall is cellulose, which is the most abundant organic compound on Earth. In fungi, it's going to be made up of chitin. That's also the structural carbohydrate or polysaccharide that we see in the exoskeleton of arthropods. If we look at the plant cell itself, the plant cell wall has a primary cell wall and a secondary cell wall. And in between there, you typically have what is referred to as the middle lamella. So here you can see the primary cell wall. Here's that secondary cell wall. Here is the middle lamella that is going to occur between two primary cell walls. So in plant cells, the primary walls are going to be laid down when the cell is still growing, and then between the walls of those adjacent plant cells is that sticky substance called the middle lamella, and that's going to glue two cells together in a plant cell. So it goes primary cell wall, middle lamella, primary cell wall of another plant cell, and then you could have a secondary cell wall, and the secondary cell wall is going to be deposited inside that primary cell wall as it is fully expanded. In animal cells, you get an extracellular matrix, or an ECM. Animal cells lack cell walls, but they secrete an elaborate mixture of glycoproteins into the space that surrounds them. Collagen may be abundant. If you remember from anatomy and physiology, for those that had anatomy and physiology, you do have those collagen fibers, and those are used for support in the extracellular matrix. They are going to form a protective layer over the cell surface, and integrins are going to link the extracellular matrix to the cell cytoskeleton, and that's going to have an influence on cell behavior. So basically what happens is that extracellular matrix is fibrous protein collagen, and the same protein found in cartilage, tendons, and ligaments. They are strong fibers that are going to be embedded in this complex web of these glycoproteins, and that's what we call proteoglycans. And those proteoglycans are going to form that protective layer over the cell surface. The extracellular matrix of some cells is attached to the plasma membrane by a third kind of glycoprotein called fibronectin, Fibronectin. Fibronectin molecules bind not only to the extracellular matrix glycoproteins, but they also are going to have proteins called integrins, and those integrin proteins are going to be an integral part of the plasma membrane and extend into the cytoplasm of the cell, where they are attached to microfilaments and intermediate filaments of the cytoskeleton. So they are going to serve as a linkage between the extracellular matrix and the cytoskeleton. And basically, since they are serving as that linkage, it's going to allow the extracellular matrix to influence cell behavior in important ways. They can alter gene expression. They can have cell migration patterns by a combination of mechanical and chemical signaling pathways. Because it's able to do this, the extracellular matrix can help coordinate the behavior of the cells in particular tissues. And we can look at that a little bit later. So here you see those collagen and elastin. Here's that integrin protein that is connecting the extracellular matrix out here into the cytoplasm of the cell. Here you can see a proteoglycan structure. There is fibronectin. So those are all parts of that cytoskeleton, extracellular matrix, so on and so forth. Those things that you should know there. This is a nice table summary of everything that we've gone through with the prokaryote, animal, and plant cell. So our two typical eukaryotic cells there, the animal and plant. I would probably write this down, make a nice copy of it in the book, and you can see those exterior structures and interior structures. Alright, so I think I'm going to stop there for this lecture. We will hit up next cell-to-cell interactions, talk about adhesive junctions, tight junctions, and pretty much end it with plasmodesmata, which are found in the plant cells. And that will take us to the end of this chapter. This chapter itself, you should know all the cell structures and their functions. You should definitely be able to tell the difference between a prokaryotic cell, a eukaryotic cell. How are they alike? How are they different? How are the two types of prokaryotes, the archaeans and the bacteria, alike and different? And how are typical eukaryotes, the plant cell and the animal cells, alike and different from one another? So there's quite a bit of information in this next test. I also would like to go over the cell membrane and its structure, and that will lead us into one of our first big labs dealing with transport across the membrane, osmosis, and diffusion. Thank you, everyone, and have a nice day.